Okay, in this unit, we're going to talk about cell division called meiosis. Meiosis is where a germ cell will divide by 2 divisions into 4 daughter cells, and those will be gametes, which are haploid. A germ cell is diploid. This happens in our testes if you're a male, your ovaries if you're a female. So the sex cells are the gametes. So egg is a female sex cell, and sperm are the male sex cells. Here we see a sperm fertilizing an egg. When an egg and a sperm fuse together, the nuclei fuse together, it forms a 2-end cell called a zygote. The 2 haploid cells fuse together to form a 2-end cell, which I call the zygote. Zygote is the very first somatic cell that we all exist as. It's a fertilized egg. There are 2 kinds of chromosomes in any kind of cell of our cells. There's autosomes and sex chromosomes. Okay, so we have 23 humans have 23 pairs of chromosomes in our diploid cells. We got 23 pairs of chromosomes, 23 homologous pairs. That means we have 46 chromosomes. Okay, of those 23 pairs of chromosomes, only 1 pair is the sex chromosomes. Okay, so 22 pairs of the 22 pairs are autosomes, non-sex chromosomes, and 1 pair are the sex chromosomes. Okay, the sex chromosomes give us our sexual characteristics. They determine our sex. There's 2 kinds of sex chromosomes, X and Y. Okay, a female sex chromosomes are 2 X chromosomes, and the male sex chromosomes is X and a broken chromosome, the Y chromosome. So we need to know that. So in our somatic cells, there's 23 pairs of chromosomes. But because it's diploid 2N, this would mean there's 46 chromosomes. But in gametes, there's no pairs because they're haploid. So gametes have only 23 chromosomes. There's no homologous chromosomes. So there's no chromosome pairs when you're a gamete. Okay, this is a picture of what's called a karyogram. This is when, during mitosis, you take a picture of a cell, all the chromosomes, and then you cut out the chromosomes and put them in order from largest to smallest, leaving the sex chromosomes at the end, number 23. Okay, so you're either going to have XX or XY, depending whether you're a male or female. Remember, XY is male and XX is female. So you see pairs of chromosomes here. So this would be a 2N cell. Chromosomes, humans. But this would be right after mitosis, because each chromosome only has one chromatid. So this would be what it looks like at telophase. Okay, all these other chromosomes, the ones that are 22, are the autosomes that contain all the other genes that our body needs besides the sex-determining genes. Okay, in a somatic cell, we have homologous chromosomes, like I just said. So you got 46 chromosomes, but you got 23 homologous pairs. Now for each pair of chromosomes, each pair, one came from mom and one came from dad. From dad's sperm, and one came from mom's egg. So remember, each chromosome has the same genes if they're in a homologous pair, but you can have variants of that gene, like eye color. Your mom could have blue eye color gene, and your dad could have a brown eye color gene. But in a homologous pair, both have the same genes, but different forms of those genes. Could have different forms of those genes. Those two chromosomes came together during the fertilization when the sperm nucleus and the egg nucleus fused to form one nucleus in the zygote. Okay, we're going to put a label, a name, on different versions of the same gene. And we're going to call that alleles. Alleles. For example, in these mice, this mouse on the left has the gene for fur color, and the mouse on the right has a gene for fur color. It's on the same chromosome. But this case, the allele for that gene is gray fur, and the allele for this same fur color gene is white fur. But it's the same. It's a gene for fur color. Same with the eyes and all kinds of other. That eye is black eye. This eye is red eye. On one of their chromosomes is a gene for eye color. But the eye color gene has different versions or alleles. So this would be the black eye allele for the eye color gene. This would be the red eye allele. So this is a nice little diagram showing the cycle, the life cycle of animals. and many plants too. So we start with a germ cell, let's say. Those are in our testes. Okay, the germ cell is 2N. You see pairs of chromosomes, homologous pairs. So the cell is diploid. Okay, so what happens during meiosis? You form 4 haploid gametes. Okay, an egg and sperm will get together during fertilization and form again a zygote. Now, the zygote will grow to be by many mitoses, 2N to 2N. It will grow to be a baby, a child, a teenager, and an adult. Okay, but in special cells, in the testes, meiosis will occur. And that's what this blue arrow is showing. One of the 2N germ cells will do meiosis and form egg and sperm, etc. So that's the life cycle. So meiosis, like I mentioned before, is two divisions that happen one after the other. Okay, so here's a germ cell. Then it undergoes meiosis 1. We call that meiosis 1. Then each of those undergoes another meiosis 2 to form the actual gametes. Okay, but in both cases you're forming, and this is a 2N cell, remember? In both cases you're forming haploid cells in both meiosis 1 and meiosis 2. Now, how's that possible? Well, let's just do the nucleus of the germ cell. So it has homologous pairs of chromosomes. And what happens in meiosis 1, you form 2 cells, but there's no pairs. The pairs have broken up during meiosis 1. So you got 2, this is unlike mitosis. You got 2 different chromosomes, but those chromosomes weren't split into chromatids. And then meiosis 2, the next stage. The next stage is splitting those 2 chromatid chromosomes into 1 chromatid chromosomes. And there's no homologous pairs there, so those are all haploid as well. Going back to the products of meiosis 1, there's no homologous pairs. You can see a pair, but they're not homologous. They're different chromosomes. So let's talk about meiosis 1. Now, both meiosis 1 and meiosis 2 have the same kind of stages, names as mitosis. So let's start with prophase. So because in meiosis 1, I call it prophase 1. So the most important thing about prophase 1 is right here. It's not a very good picture. The homologous chromosomes get close to each other and they touch each other and they exchange pieces of each other. It's like they're hugging each other because they're going to say goodbye, and they're giving each other souvenirs of themselves. So this is called a tetrad, when two chromosomes in a homologous pair touch each other. Okay, so let's say this is from mom, the red one's from mom, and the dark blue one's from dad. What happens in the tetrad is, like I said, they exchange pieces. So let's say A stands for black eyes, and little a stands for red eyes. So what's happening in, let's say blue is dad, is the gene that you got from your dad, and red or pink is the gene you got from mom. Okay, so mom has, let's see, dad has big A, so he has black eyes. And your mom had red eyes. So what's happening at prophase 1, when the tetrads are formed, that means when the homologous chromosomes pair up with each other in each pair, they exchange pieces. That's called crossing over or recombination. And as they cross over, genetic recombination, as they cross over, they exchange pieces, so that in your in your germ cell, now you have a new mixture of alleles in each gene where a crossover, in each chromosome where a crossover occurs. So these chromosomes, it's not just one crossover event. There's all kinds of, depending, there could be a lot of genes being exchanged. I should say gene alleles being exchanged. Okay, so this adds to, this is why the children of a mother and father have a different mixture of alleles on their chromosomes than the parents do. This adds to the genetic variation in the kids. Sexual reproduction does this. Asexual reproduction will have no crossing over during prophase or any time. Mitosis does not have crossing over occurring. The daughter cells formed by mitosis have the same alleles as combinations as the mother cell had. So the rest is pretty basic. At metaphase 1, which is metaphase or prophase, the chromosomes line up randomly at the metaphase plate. Actually, the tetrads line up, but recombination has stopped by now. Remember, crossover only occurs during prophase. Now, they're trying to make an important point here. The way the chromosomes line up during metaphase 1 is random. That means one side isn't, like the left side isn't just mom's chromosome and the right side's dad's. No, the two chromosomes in a tetrad, mom's or dad's, your mom's or your dad's, are randomly mixed up. So this could be mom's on the left side, this could be dad's on the left side. And that's important because that's another source of variation called independent assortment. The genes are the alleles are independently assorted. So genetic variation in the offspring is because these two things: crossover, which mixes up the cards in the deck, as it were, and independent assortment, which would be the chromosomes lining up randomly at the metaphase plate. So if we do a little statistics, how many different varieties can we have the chromosomes? If you have 23 chromosomes, how many different ways could you, patterns could you have, mix-ups? So, because there's two chromosomes, each homologous pair, and you bring that to the 23rd power because there's 23 pairs, that'd be about 8 million ways of combining the homologs, so that 8 million different ways. So sometimes the mother will be on that side and the father chromosome will be on the right side, or it might be on the left side, right side. And you do, with 23 different pairs, do the math, and you'll get 8 million different combinations. That's called independent assortment. And because both egg and the sperm are doing it, you multiply those two probabilities. 8 million times 8 million is 64 trillion. So there's 64 trillion combinations in the zygote, just because of independent assortment, not even including crossover in prophase 1. Okay, telophase is simply you form, the cell forms new nuclear membranes around each of those new nuclei. Remember, the nucleus is haploid because there's no more pairs of chromosomes. There's chromatid pairs, but those aren't homologous pairs. So you only see singletons. You don't see any This is not a homologous pair with this guy. So it's 2N. Whereas over here, you had, in the tetrads, you had a 2N cell. Nucleus, a 2N nucleus, whatever. Okay, after that, cytokinesis occurs. The cytoplasm divides, but it's called interkinesis because same thing as cytokinesis, except that the cell division isn't over. You still have meiosis 2 to do. So meiosis 2 is very much like mitosis. I mean, there's no crossover event between homologous chromosomes because there are no homologous chromosomes anyway. Meiosis 1, remember, started with one diploid germ cell. Okay, so it was a 2N cell and finished with 2 haploid daughter cells. So, each of those haploid cells will divide in meiosis 2 to 2 daughter cells. So again, meiosis 1 is that 2N to N, and then each of these guys will divide to an N cell as well, haploid, except there won't be any 2 chromatid chromosomes in these gametes. So let's look at meiosis 2. So you're starting out with no homologous chromosomes, but you have 2 chromatid chromosomes. Because in meiosis 1, the homologous chromosome pairs were broken up, but no 2 chromatid chromosomes were split, like in mitosis. So, all we have to do is look at one of these, but it happens with the other daughter cell as well from meiosis 1. Okay, so what's going to happen? Metaphase, that's prophase. The nuclear membrane disappears. Spindle forms, moves the chromosomes to the middle of the cell. The metaphase 2 means happens during the second meiotic phase stage. So the 2 chromatid chromosomes line up at the equator of the cell, pushed by the spindle or pulled by the spindle. The next stage is anaphase 2. In anaphase 2, each 2 chromatid chromosome will split into 1 chromatid chromosomes. So you can see, like, the X is breaking up into 2 chromatids. The 2 chromatid chromosome is breaking up into 2 1 chromatid chromosomes. Okay, anaphase means the point that they're the stage where they're separating and moving to opposite sides of the cell. Once they hit the opposite sides of the cell, the nuclear envelope and the nucleolus starts appearing again, and you'll get cytokinesis happening where the cells are cleaved down the middle. Okay, so again, 4 haploid daughter cells are formed from 2 haploid, 2 chromatid chromosome daughter cells from the first meiosis stage. Okay, here's some review questions. 2 chromosomes in nucleus that carry, this means location, of a specific gene. So 2 chromosomes in a nucleus that carry locations of a gene for the same traits in the same positions on the chromosome, of course. That means they're the exact same loci, but can specify different versions of the same trait, constitute a pair of homologous chromosomes. Okay, so if they had the same exact gene, even though it's a different version, these would have to be homologous chromosomes. Here's kind of a silly question, but it kind of makes sense. A karyotype. Okay, that's like a lineup of all the chromosomes in a cell, paired of the homologous chromosome pairs. So if this is a human karyotype, the karyotype would have 23 pairs of homologous chromosomes. So this would be like a picture of every couple at the prom all lined up next to each other. Okay, genetic rearrangement of the genes between 2 homologous chromosomes. That happens during prophase 1 of meiosis, is called crossing over. Okay, I didn't mention this, but when you cross over, when 2 homologous chromosomes touch and cross over, it's just not they're exchanging genes. But it's not just one gene like the picture showed, a few genes. It could be many genes, it could be a few genes, depending on how big of a segment of the homologous chromosome is traded off to the other homologous chromosome. So this is true. It could occur many times simultaneously on a homologous pair during prophase 1. This is kind of a tricky question, but I took some time to explain it before. Independent assortment means when the homologous chromosomes line up during metaphase 1. Say here's some more. One's from, okay, let's say this is your germ cell this is happening in. Okay, this is from this chromosome you got from your mom's egg when you were a zygote, and this you got from your dad's egg, sperm. And so the way they line up is totally random. So that could be dad's on the left side and mom's on the right side. And now there's 23 homologous pairs, at least for human chromosomes. So it's like shuffling a deck of 23 cards. Actually 46 cards, because there's 46 chromosomes. And which one ends up at the metaphase plate, which one ends up on what side in the homologous pair is totally random. So that's called independent assortment. So which of these is a false statement? Meiosis occurs in the ovaries and testes of animals. Well, that's totally true. That's the only place meiosis occurs, in animals. Sexual life cycles involve an alternation of diploid and haploid stages. Let's see some other ones. Mitosis produces daughter cells with half the number of chromosomes as a parent cell. No, mitosis is 2N to 2N. A normal human has 46 chromosomes. That's true. This guy is false right here. So letter C is the right choice. Let's go back to letter B. What do I mean? So in our life cycle, that's in our from formation of a zygote to your whole life. When you're making sex cells and somatic cells, so we start as a zygote, which is 2N diploid, and then the zygote breaks up by mitosis, doesn't break up, divides many times by many mitoses until you're a person. Until you're a baby, then a toddler, and then a preschooler, then an adult, then an old person. Okay, so mitosis keeps on, allows the organism to grow and repair dead cells. Okay, so that's 2N to 2N. Many times over. Now, some of those 2N cells in the ovaries and testes will do meiosis to form eventually a gamete, which is haploid. Haploid as opposed to diploid. Okay, so that's happening right here. Okay, if you have sex with the opposite sex, gamete plus another gamete from another person will do fertilization. That's a process we call fertilization, where we have N plus N forms 2N. So, our sexual life cycles, our life cycle involves alternation between diploid and haploid. That's what alternation means. There's a switching between diploid and haploid cells. Now, the gametes are haploid, and all the rest of our cells are diploid, including the very first cell that we started out with. So we have diploid and haploid parts, phases of our existence. We once existed as a sperm and an egg. That was us before not really until they unite in the process of fertilization, but we were haploid before we existed, I guess. And then, of course, we produce haploid cells in our sex organs, which does not occur during meiosis. Crossing over, yeah, it does. It occurs, so that's not the right answer. Pairing of homologous chromosomes, yeah, during prophase 1. Random fertilization, fertilization happens after meiosis once you have a gamete, so that's not it. Production of gametes, yeah, the whole point of meiosis is to produce gametes, so the false one is letter C. No, identical twins came from one zygote. I didn't tell you this, but one zygote is 2N. Then it forms 2 by mitosis. It forms some more 2N cells. But those 2N cells usually stick together. Normally stick together after they're formed. They connect to each other, and it continues like that until you have embryo with many cells stuck together. Now, at some point, by accident, we don't know exactly how, um, those can get separated. Those, and it's just not when it's at the 4 cell stage, it can be at any stage, but they get separated early on. And each of these is 2n, 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 2n. But when they do, you got 2 new individuals, which are identical genetically, because they came from the same sperm and egg. From the same sperm and egg. That's fertilization. So now if you have fraternal twins, that means more than one egg got fertilized by different sperm. So there's different rearrangement of the genes in the 2 zygotes because those genes were mixed up by independent reassortment and crossover when the egg and sperm were formed. So you have different egg and sperm. You have different combination of versions of the genes in each sex cell that's produced. So we're talking about normal brothers and sisters, not twins or fraternal twins. They might look similar, but they're not identical because no, they don't have. They have identical chromosomes in the sense that they share all the genes that the brother or sister has, but they have different versions of the genes, different alleles, same genes, different versions of the genes, which we call alleles. So that one's wrong. They have identical genes, but not chromosomes. That just does not make sense. Kind of does, but it's very vague. They have similar but identical, but not identical combination of alleles. Okay, so if they had changed that to alleles, that would be the right answer. They have a small chance of having identical genes. That would make them look. That doesn't make sense. Okay, so letter C is the best, even though it's kind of poorly written. They should have crossed out the word genes in this question too. Okay? So then definitely C is right. Some accidents can happen during meiosis, and usually, sometimes these accidents produce gametes that still survive, but cause problems once the zygote is formed. So in meiosis 1, what can happen is that the twins, the homologous chromosomes don't separate. So the homologs, that's a short version, abbreviation. That's a single word for the word homologous chromosomes. The homologs, some homologs don't separate at anaphase 1. Okay, so they don't want to say goodbye. They stick together and they go off. So that one guy has 3. Okay, one guy gets, let's see, 1, 2, 3 chromosomes, when they split during anaphase 2, one only gets 2, one only gets 1. I mean, so this is like, this first one is ana 1, and then this would be ana 2. But you can see that there wasn't a mistake. There was no sticking in ana 2 as the mistake was in anaphase 1. And you'll get one extra chromosome for each one that's stuck up there in 50% of the gametes, and you'll have one less chromosome in 50% of the gametes that form. This usually does not, if that egg or sperm gets fertilized, usually it doesn't go on to make a healthy embryo, and the embryo dies. Now, you could have a sticking together during anaphase 2. Let's say, yeah, the pairs separate, okay, but what happens is that now the X did not separate like it should have into those two pieces during. It didn't split. The 2 chromatid chromosome did not split into 2 chromosomes. One did not split in the middle of the 2. That's very much like what happens in mitosis normally, but that should happen in meiosis too, right here. But it doesn't, and you'll get the effect of having these split. You'll have 3 chromosomes. You have extra chromosome that you don't need. You'll be missing a chromosome, and then but you'll get some normal. You'll get some normal haploid gametes. 50% of your gametes will be defective. Of those 4 that are formed, 2 will be defective. I forgot to mention that the sticking of chromosomes during meiosis, either during anaphase 1 or anaphase 2, if they stick, this is called non-disjunction. Disjunction means separation. So, non-separation. Okay, so separating, this means the chromosomes are not separating like they should. In meiosis 1, the homologous chromosomes aren't separating. In meiosis 2, the chromatids aren't separating during anaphase 2. In most cases, again, any embryos that develop from zygotes that had one of the gametes, having either too many chromosomes or too few, will probably not allow the embryo to develop. And at an early stage, it will abort spontaneously. The mother will lose that embryo. We call that a miscarriage. Okay, non-disjunction causes a situation we call aneuploidy. Okay, that causes the zygote to have one X chromosome or one fewer chromosome than normal. Okay, we'll talk about polyploidy later. One common type of aneuploidy is trisomy, where you have an extra copy of one chromosome. If you're the zygote that's missing one chromosome, then that aneuploidy is called mono or one-somy, monosomy. Down syndrome is an example of trisomy. In a karyogram, chromosome number 21 has an extra chromosome in the diploid state. So trisomy is an example of aneuploidy, and it's caused by non-disjunction mistakes in meiosis in women that are 40 years or older, usually, when their eggs get fertilized. The risk gets bigger as the egg gets older. Okay, so if you have non-disjunction in the sex chromosomes, if it's the Y chromosome that you have more or one more or missing one, Y chromosome carries fewer genes because of its size. So that kind of mistake won't be as obvious in the individual that grows from the zygote. But if you have more X chromosomes or fewer missing the X chromosome totally, then there's going to be issues. There's going to be bigger issues, health issues, for the individual who gets that. Also, there'll be fertility issues. You might be sterile if you have aneuploidy of the sex chromosomes. This is an example of aneuploidy with the sex chromosomes. It's called Klinefelter's syndrome. This person would look like a male, but he has an extra X chromosome. A person with Klinefelter syndrome, as he grows up, might not produce enough testosterone, the male hormone, and that would cause him to be maybe infertile. His sperm wouldn't be able to fertilize an egg. I mean, it would fertilize it, but it wouldn't make a viable living embryo from the zygote. Lacking testosterone would probably cause you not to have very much facial hair. It would cause you to maybe develop breast. So, but a lot of these people, you can't even tell they have Klinefelters. It's about 1 in 750 males have it. Okay, Turner syndrome. If you have Turner syndrome, you only have one X chromosome. So you are a female because you're totally missing the Y. You don't have the Y chromosome, but you're missing an X chromosome. So you're gonna be infertile unless you get estrogen supplements, the hormone, the female hormone. But otherwise, you're normal for the most part. Now, some cases you have heart disease early on. There's problems with the valves in your heart, and other things. You're usually really kind of short in stature, but otherwise your intelligence isn't affected. Polyploidy is when you get an extra full set of chromosomes. So it's not one extra chromosome or one less chromosome. It's a whole homologous set of chromosomes. This could happen if two diploid cells were to fuse, like an egg and sperm fuse. What if two diploid cells somehow fuse? This would produce polyploidy. Or what if a diploid cell fused with a cell, egg or sperm cell that only got through the first stage of meiosis? Then that could happen too. You could get a polyploid cell. Now, if this happened with humans or animals, this is lethal. The zygote would never live past a few cells, divisions. Now, but for fruit, you can increase the fruit yield, or vegetables. You can make bigger fruits by making the cells artificially, making them polyploid, triploid, or tetraploid, or even higher flowers too. You get bigger flowers. Okay, so let's do some review questions. Non-disjunction occurs when chromosomes don't disconnect, when they don't disconnect. Usually because of a mistake in anaphase 1 or anaphase 2 of meiosis. So a portion of a chromosome breaks off? No. Chromosomes replicate too many times? No. Two chromosomes fuse into one? Not really. The chromatids of a chromosome pair, well, the members of a chromosome pair fail to separate. That's true. Or the chromatids fail to separate. That would be true too. Okay, aneuploidy means missing one or having an extra chromosome in a homologous pair. So usually is a problem in meiosis, 1 or 2. So it happens in meiosis when you get fewer or extra. Okay, so D is better than C, because you can have fewer, and that would lead to monosomy, like Turner's syndrome, or trisomy, like Down syndrome, or Klinefelter's syndrome. Individual with trisomy has extra copy or copies of a chromosome. You got one extra copy of a chromosome. You got 3 chromosomes instead of 1. Yeah, if you have polyploidy, you won't even survive the embryo stage. So there's no way you can reproduce. So that's a false statement.